All right, so welcome everyone. Today is lecture 18, and our guest speaker today is Wengong Jin. So he's actually one of the pioneers, I would say, of this new field of using graph neural networks for designing new drugs and new therapeutics. And he will actually give the whole lecture. So this is a little unusual for our class, but he's graciously agreed to not only introduce us to his own work, but to also introduce us to the general field and sort of what came before and a little bit of his outlook. So Wengong, take it away.
All right, let me pull up my screen here. Okay, yeah, it's great to be here. Thanks for inviting me. Yeah, hello everyone. Today I will talk about some of my work on deep learning for drug discovery. This is something that I've been excited about for in the past 4 years, my PhD. It's my pleasure to introduce some of the work that I have done in this space. Drug discovery is a very time-consuming process. It takes more than like 10 years for a pharmaceutical researcher to actually identify a new drug from like millions of drug candidates. So the average time and cost for designing a drug is actually around like 10 years, and the cost is around like $2.6 billion. This is just some data that I copied from the Pharmaceutical Research of America. So obviously, we don't want to wait for another 10 years, like getting locked down at home, just waiting for a COVID drug to come out, right? So we do want to find drugs very quickly in order to respond to this pandemics or novel pathogens. In the computational terms, drug discovery is a search problem, and you can think of this chemical space as some sort of maze, and you want to find a good drug that can kill the virus or the pathogen. And this is sort of the central goal of this of drug discovery. But it turned out that this computational task is very, very challenging. And the challenge comes from the size of the chemical space. So the number of drug-like molecules is estimated to be around like 10 to the 60th. People give different numbers if you ask different people, but it's usually around 10 to the 30s to 60s. And believe it or not, there's no way to brute force screen all of them. Even in industries, these pharmaceutical companies, standard experimental, like high throughput screening methods can only test around like 10 to the 5th compounds, like it's 100,000 compounds per day. So compared to this astronomical number, this is really nothing. So instead of doing this kind of brute force search, we should develop some efficient algorithms to design drugs automatically rather than just putting all of these compounds into your wet labs and then do experiments. So, and this is sort of the major goal. And that's where like deep learning or artificial intelligence comes in. So let me introduce to you like this field and some like basic concepts. So the community has been looking for a lot of different kinds of solutions, all based on computation. So here there are two important concepts. One is called this functional space. Basically, what's the property of each compound? The mapping from the compounds to the functional properties, like toxicity. That's one classic property which you don't want in your drugs. There's also solubility constraints, and there are also other properties. So these properties are usually measured by you can just give a numerical score, like if the toxicity is around like 10, then it's pretty bad, and the solubility is around like 5, then it's good, something like that. The second concept is this chemical space that I briefly mentioned. It's basically this gigantic set of potential molecules. So the goal is to find like a chemical structure that has particular set of properties. And there are different, so in the past, so briefly, it has like 3 different types of approaches to find drugs with specific properties. One is based on simulation. So this could be like molecular dynamics or molecular docking, for example. These simulation is based on first principles, like how basically based on like the force field, how the molecule wiggles around in the space in a protein pocket, for example. Then you would know whether this molecule binds this protein. The second type of approach is called virtual screening. So basically what you do is instead of doing that simulation, you directly predict, like if I give you a structure of the molecule, can you directly predict its solubility or toxicity or etc. And then if you can predict that well, then I can actually put a lot of these candidates into your virtual screening model and then just select the top ones that has the best solubility or the lowest toxicity. But this is still kind of stupid, right? Because you still can't put all the 10 to the 60th compounds into your model. That will still take like forever. So the last approach is called de novo drug design. It's the most ambitious among the 3, so it basically wants to solve this inverse problem by looking at the set of criteria like low toxicity, high solubility, and then can you reverse engineer the right compound from this set of criteria.
So Wengong, can I ask you,
yeah,
where does the precise docking constraint comes in? Because right now you're talking about properties that are just simply general properties, but how do you now get it into a functional site of a protein, for example?
Yeah. So you can measure, give a numerical score of the binding affinity, right? Basically for each molecule and given a protein, what's the binding affinity? And that's that's basically PK scores, which you can just give. It's basically a numerical score.
So what you're basically saying is that the docking is a whole orthogonal problem to this. And basically, there's 2 problems. On one hand, you want the binding affinity of a chemical to a protein. On the other hand, you want certain properties of this molecule which are independent of that?
So I would classify docking into this simulation-based approach. Can you see my mouse?
Yeah. Your mouse.
Yeah. So docking is usually based on these like force field, right? These energies, how our energy functions, what's the low. Basically, molecular docking search for the lowest conformational, the low energy conformation inside of a protein pocket by doing this multi-color simulation based on the force field. And then based on that, you can try to figure out what's the binding affinity. And that is a very expensive step, right? Because you need to do all these simulations.
Yeah.
Virtual screening is try to bypass that by just looking at the structure. Can I predict the binding affinity score, which is just a numerical number?
And the arrow, however, in your second column is pointing down from the virtual screening to the chemicals. So that means that if I know the properties, I can actually infer particular chemical structures. I can.
Well, these are some. So this is the figure that I copied from this paper. I think what I want is actually the arrows pointing from the bottom to the up.
Got it. No, I agree with you. That's
right.
I would have drawn them as well.
Oh yeah, I missed this. Yeah, should have fixed that. But
okay.
Yeah, sounds good. So the last one is the arrow points from the functional space to the chemical space.
That I agree with. Problem. The third arrow I agree with. Perfect. Thank you.
Great. So obviously the simulation is often too slow because it takes around like days, or if you want really very accurate simulation, then it will take days for just for one compound. So I will kind of omit that in today's talk. And I will focus on the second and third approach. So virtual screening is basically, like I said, here it's just more elaborate. I just want to elaborate what I really mean by virtual screening. So basically, the input to this virtual screening model is a compound, and then I can predict some numerical score, whether it is good or not. And that depends on what is the end property. Like if it's binding affinity, then we need to predict whether this compound has low binding affinity. If it's toxicity, then it's basically the same. And virtual screening is favorable because it is much faster than experimental screening in wet labs. It can test around like 10 to the 8th compounds within a day, while experimental screening, it will take more than 1 year. And also it is quite cheap because everything is in the computer rather than in the wet labs. But virtual screening also has this trade-off. So on the right, I plotted this. I have two axes. One is the coverage of the chemical space, and the y-axis is the ease for synthesis, which represents how fast you can actually test a compound in the wet lab. Because in the end of the day, after like your virtual screening method propose some compounds, you still need to validate them in the wet labs. So virtual screening limits itself within the set of commercially available compounds. For example, like the zinc library. Basically, you put all these commercially available compounds in to your model, and then this virtual screening method gives you a rank over these candidates, and then you just test the top ones in your wet lab. So the advantage is there's no need for like synthesis of any compound. So after you generate the ranking, the test is relatively easy because you don't need to synthesize them. But obviously it loses the coverage because at best we can screen up to like 10 to the 9th, 10 to the 10th, still not comparable to the entire chemical space. And also I will talk later in the later slides, these traditional techniques, traditional virtual screening techniques are based on like handcrafted features, which limits its accuracy. So in a way, the model often produces wrong predictions that in the end, it does not result in good. It's actually not good when you test these compounds in wet labs. So the second paradigm that I mentioned is this de novo drug design. It's the inverse to the virtual screening. Basically, from a set of criteria like potency, safety, reverse engineer what's the right drug or right compound. And that's a very challenging problem because molecules are, this structure, and standard like machine learning models that we typically encounter, the output is just a numerical number rather than some sort of structure. So that's why this problem is much harder than virtual screening. De novo drug design also has this trade-off between the ease for synthesis versus coverage. So it certainly can explore the entire chemical space very efficiently because you don't need to try every compound. The model will figure out that for you. But the model will actually suggest some pretty new compounds that's not commercially available. So you actually need a synthetic chemist to learn to figure out how to synthesize that compound in order to test them in wet labs. So that slows down the experimental workflow, but it gives you more potential in discovering something new. Oh, sorry. So I also forgot to mention that this problem traditionally is based on these genetic algorithms by doing some sort of random mutation on the molecules. And then these mutations are sort of time design rules. So in a way, we don't have yet efficient algorithms to explore this chemical space efficiently. So in a way, even though de novo drug design or virtual screening, the idea has been there for over like 2 decades, but it's actually quite hard to make them actually work.
Can I ask you another question? So is it feasible, basically, how much is this done of basically saying, listen, this space of this part of the chemical space, I know nothing about. So let's synthesize a bunch of molecules to explore that part of the space. Namely, when computationally you're sort of saying, oh, this type of part of the chemical space seems interesting, but I know nothing about it. Can you somehow couple the experimental exploration for seeding and then the computational search, or is that not really done?
So what you can do is actually distill the experimental data. So basically, people have done like wet lab experiments in the past, right? So then you have these molecules and their like properties labeled. So you can actually distill a scoring function from this set of experimental measured data points, and you can use that scoring function to guide your this de novo drug design, like the search algorithm.
But I'm thinking about the converse, where your de novo can now basically say, well, I would love to test all of these molecules, or hey, there's a property, there's a class of molecules that no one has tested before. Can you go and test a bunch of those to sort of expand the training function?
Yes, you can. So that's definitely, you can also update your scoring function after you have tested the compounds. And that's more like a closed loop. That's the most ambitious moonshot, right? Of this closed loop system. But the difficulty here was, sorry, go ahead.
No, where you're synthesizing tens of thousands of compounds in combinatorial ways to explore the space specifically for the purpose of training.
Yes, that's totally possible.
Okay. Yeah. So I'm sorry for asking so many questions. Please carry on. You have so much to cover.
Sure. Please feel free to ask any questions if you have. I think the time is sufficient for now, based on my estimation. So I keep mentioning that these traditional techniques are not efficient or not perfect, and let me explain why this is the case. Nowadays, there are a lot of research of interest in this computational drug discovery, primarily because of the rise of deep learning. So in computer vision, like deep learning has achieved like human level accuracy in computer vision. And the key to success is actually this automatic feature learning, right? When I mentioned about like virtual screening, traditional methods are based on these handcrafted features, which inherently limits the performance. I would elaborate this more in a case study later. But what deep learning can do is to learn these features automatically for you based on the data that you have. So in a way, you don't have to hand design these features, which may be the bottleneck of the model performance, but rather ask the model to learn these features for you based on the data end to end. So that's one way why deep learning, a lot of people got excited in applying deep learning for drug discovery. On the other hand, if you look at this de novo drug design problem, it's traditionally based on these hand design rules. But in deep learning, there's a type of model called deep generative models, which can generate these realistic complex objects, right? Like images or text. This is just one example that I pulled up from a paper called DALL-E, which is an image captioning, image synthesis model. You have a sentence say, I want to generate an image of an armchair in the shape of avocado, right? And the model can actually generate images quite well that sort of fits your description. So coming back to this de novo drug design, now I can actually use deep generative models to generate compound with desired properties using these deep generative models. So this allows you to this no longer requires these hand-designed rules by how to mutate a molecule, but the model can learn how to generate them and also cater them into with respect to specific properties. So that's sort of why deep learning give a more like revolutionized this field. And the main technique that I will talk about today is based on graph neural networks. So if you look at the two problems, this virtual screening, other people call it molecular property prediction, and also the problem of de novo drug design. It's basically this loop, right? From the molecule to the property, and then from property to the structure. And the reason why graph neural networks become useful is because molecules can be just modeled as graphs. So for example, each atom in this molecule can be is a node in this graph, and each edge is each bound is an edge in this graph. So now the problem of like virtual screening is basically learn how to encode this graph into some sort of representation, then based on that representation predict the right property. And this de novo drug design problem is basically this graph generation task. Well, given a set of criteria, generate the right graph using the deep generative models that I talked about. So just as an example of how these what these can do, our lab, through collaboration with the Broad Institute, we actually discovered a powerful antibiotic called helicin, which can kill a lot of bacteria which are actually resistant to existing antibiotics. So this is sort of the just want to show some examples of what we can actually achieve using these graph neural networks that the previous methods cannot achieve. And I hope that you will feel excited about this field. So this is sort of a long sort of introduction of what the field is. And today I would like to actually give you like three sort of case studies on like what sort of techniques what are graph neural networks, how to use them to discover new antibiotics or COVID-19 drug combinations. There are actually a lot of topics that I feel is important. For example, how we can incorporate like biological knowledge into these neural network architectures, and also how to do graph generation. Graph generation is actually a pretty new problem in the context of deep learning that people can generate images or sentences, but not really graphs. So that's in the third part, I will kind of talk about some new techniques for like graph generation, and actually that makes made a lot of excitement among the machine learning communities. So let's dive into the first part on antibiotic discovery. So let me explain why we got interested in this antibiotic discovery. This figure gives you sort of the history of discovered antibiotics. Since 1930s, we discovered penicillin, and it has been quite fruitful over many decades. But after the low-hanging fruits were gone, it started to become really hard to discover novel antibiotics. But on the other hand, because we sort of abused these antibiotics a lot, these bacteria actually became resistant to many antibiotics on the market, in the clinics, and that's why it became a huge problem, because in many of the deaths in the hospitals are actually caused by these bacterial infections, and there's really no cure because of antibiotic resistance. So we need to find novel antibiotics to fight against these resistant bacteria. So I want to use this antibiotic discovery project to sort of illustrate how virtual screening works and how to use deep learning to do accurately to do this virtual screening more accurately. So what we did was with collaboration. So we had a collaboration with biologists in the Broad Institute with Professor Jim Kellis. We collected around like 2,500 molecules with measured growth inhibition against this bacteria called E. coli. And we collected, so each molecule is labeled as positive if it inhibits this bacteria. So you see on the table here, I have a bunch of drugs, and then whether it is antibacterial or not, then I can use this experimental data to train a model to predict whether a new compound will kill this bacteria or not. So, and this model is called graph neural network. And let me explain let me briefly introduce what graph neural networks are, and then why also I will also explain why we choose graph neural network over other models.
So Wengong, we had a lecture on graph neural networks, but this will be a wonderful review. So assume that they've seen it once, and then you can get technical on us.
Sure, sounds good. So let me first explain why we wanted to use graph neural networks. So the traditional approach in this virtual screening realm are based on these fixed hand-designed molecular features. For example, like molecular weight or number of heavy atoms. There are also more sophisticated features called like Morgan fingerprint. What it does is quite simple. So as you can see from the figure on the right, what it does is basically like chop your molecule into different substructures, like a benzene here, and then I can map them into a sort of a bag of substructures. Each entry represents like here it is 1 because this molecule has a benzene, and so on. So you can get in this way you can turn a molecule into a vector representation. So what you need to do is actually even though conceptually simple, but in practice it's actually quite intensive because you need to sort of enumerate possible like substructures up to like radius 2 or 3, and it actually because molecules are combinatorial, right? So you actually have a lot of these possible substructures. So in the end, what you get is a very high dimensional feature. In the case of Morgan fingerprint, the typical dimension is around 2048, and different substructures actually has to be merged by some sort of hash function, because you just if you don't do this hash function, you'll get like more than 1 million size vector. So in a way, this gives you a representation that is sort of funny in a way because each entry also represents different types of substructures. So like I said, these features are designed by experts, like these Morgan fingerprints. And then after you turn that molecule into a feature, then I can do some sort of model on top, right? This is what the traditional method does, like support vector machines, random forests, and so on. And that's sort of the typical workflow that has been there for more than decades. But but the problem is these features designed by these experts do not necessarily capture all the antibacterial patterns. And believe it or not, we don't really know what are these antibacterial patterns. If we know them, we would have discovered all the antibiotics already, right? So in a way, this hand-designed features can miss a lot of these unknown patterns because the way that how we engineer these Morgan fingerprints are sort of agnostic to the end properties, right? So that's why we decided to use graph neural networks. and the goal is to sort of learn these features jointly with the model. So now the feature and the model become one, and these graph neural networks can learn these feature representation directly from data, and this representation is learned in a way that captures these antibacterial patterns. So now let me just briefly go, I have just a quick animation of what graph neural networks looks like. As a disclaimer, I'm not inventor of graph neural networks. GNNs is a standard deep learning model that has been proposed by many researchers. The input to the model is, to this neural network, is a graph. And so we need to represent each molecule as a graph where each atom is a node and each bond is an edge, right? I hope this is straightforward. Now, given this graph structure, I need to sort of turn that into a continuous feature vector, right? None of the models can directly operate on graphs. So this procedure is called graph convolution. So initially, let's look at just these 4 atoms here. Initially, each atom has a vector that indicates its atom type, like whether it is a nitrogen or a carbon. So now when I have these 4 vectors, so now I can apply this graph convolution. It is very similar to convolution in images. You just squash these 4 vectors into a single vector by a linear layer, right? Followed by some nonlinear activation like ReLU or sigmoid. Now, these 4 vectors get squashed into one, and this vector actually encodes a local subgraph. Looks like this, right? Because it sort of came from these 4 vectors that each represents one atom. So I can actually apply this procedure many times. If I add one more graph convolution afterwards, then it actually encodes a larger subgraph, right? And the good thing about these graph neural networks is it's all continuous. So I'm not constructing this long, high-dimensional feature vector, but rather we're condensing everything into this low-dimensional continuous space. And this allows you to, because the feature now is low-dimensional, you can actually learn, generalize better to new data sets and to new molecules. So at the end, we apply some sort of pooling operation that combines all these vectors into a single vector that just summarizes what this feature, what this graph looks like. I can then add some sort of feed-forward network on top of this feature representation learned by this GNN to predict a single numerical score, like basically a probability, how likely it can kill the bacteria. So again, pulling back this paradigm, basically traditional approach use this handcrafted feature and then learn something on top of this feature vector. But now we have these 2 steps learned jointly within this graph neural network architecture. Okay, so the model, I will kind of skim some of the experiments that we did. So first, we wanted to evaluate what's the performance, like predictive performance on a holdout test. And the model actually can do pretty well around like 0.9 AUC on the test set of like on the molecules that we experimentally measured their antibacterial properties. So after we finished the model training and testing, then we actually used it to rank around 10,000 compounds. It's not big. It's coming from this broad drug repurposing hub, where these molecules are already like they are drugs, but they're used in other contexts, not as antibiotics. So we ranked these compounds and see whether we can actually repurpose these drugs for our use of like antibiotics. And believe it or not, we actually discovered a new compound called halicin originally it's labeled as SU3327, and it is structurally novel compared to the existing antibiotics. And as you can see here on the right, this molecule has low molecular similarity, has a low similarity to the existing antibiotics. And this is a really good news to us because we are looking for something new because existing antibiotics are already suffocated. Their bacteria are not effective against these bacteria anymore. So we want to find something new that may not suffer from the same problem.
Wengong, can you explain a little bit what you mean when you say that you discover these compounds? So basically they're and that it's novel. So there's libraries of compounds, but this compound was previously synthesized. It was just not known to have antibiotic properties.
Yeah, it is not known to have antibiotic properties. It is originally used as a JNK inhibitor, basically a kinase inhibitor, but it is not used as an antibiotic before.
Okay, perfect. So you're not yet you're not yet generating new compounds, you're just testing compounds.
Yeah, I shouldn't use the word discover, but we kind of rediscovered the new use of halicin.
Exactly. And then you named it, or they had already named it?
We named it.
Okay.
Before it didn't have a name because I guess it's not really used in the clinics.
And explain the origin of the name.
It came from Star Trek, I believe. That I forgot my collaborator named it, but it's from a novel called The Space Odyssey. That's what I remember.
Very cool. That's what I'm glad you shared the story. That's wonderful.
Sounds good. Yeah. So, I only showed that halicin is novel compared to the existing antibiotics, but it is also very effective. So first of all, it showed a potent growth inhibition against this bacteria called E. coli. So here I have this response curve. The y-axis is the optical density. Basically, the lower the better. Like the lower means like the bacteria are all getting killed. And yeah, sorry, I have this. I kind of missed, kind of added this in the last minute. So I have this figure here again. But it is also effective against these resistant bacteria strains in mice. Which is quite surprising because we only trained our model on this bacteria called E. coli, and we discovered this halicin. And this halicin is actually has a broad spectrum antibacterial properties. It can kill other bacteria that's known to be resistant. So, for example, this Amiba is a notorious bacteria that's pan-resistant. Pan-resistant means it's resistant across every antibiotic family. So it is really nasty, but halicin is able to kill the bacteria.
This is amazing result. So are you able to now go back and see what could you have done with previous representations and maybe expand the previous representations? Basically, have you sort of said what aspect of my representation learning was the most crucial? And more generally, have you gone back and asked, are there whole classes of representations that I can now go and augment previous models with?
Yeah, that's a good question. I don't have a good answer for it because we in a way we still have not yet known like what's the new representation that sort of facilitated the discovery of halicin. We will kind of do this in the future. Yeah.
Okay, great. Thank you.
Yeah. On the right, we also see that that this bacteria called C. difficile is also resistant to many antibiotics or existing antibiotics. For example, if you treat this bacteria with metronidazole, and this drug is the frontline, the first choice in treating this bacterial infection, but it is actually not effective anymore because of the antibiotic resistance. And again, the infection is gone if you treat it with halicin. And these numbers are all like done in mice, right? So it's in vivo rather than just in cell-based assays. So in a way, it is a lot more realistic measurements than we typically have. And these are very exciting results.
Am I reading this graph right? Vehicle is actually doing better than metronidazole. Does that mean that basically the bacteria have now evolved to actually enjoy the drug that they used to try to kill them with?
At the one for after 6 days, it seems that I think there are some bunch of red points that's in here, which it's kind of just like blocked by these blue dots. Blue dots represent halicin. Red dots are this metronidazole, and so it still does better than the vehicles. Yeah.
No, but what I'm trying to point out is that metronidazole actually does worse than the vehicle. Like the red is actually higher than the green.
Yeah, but there are a bunch of red points that's in here. That's why it seems very.
Got it. So they can evolve resistance to it easily, but for a big chunk of them, they're actually dead.
For certain chunk of them, they're dead. Yes.
Yeah. Perfect. Yeah. Thank you. Again, these results are amazing.
Okay, thanks. So, let's see here. Yeah. So we also wanted to kind of, so the model is we apply this model on just 10,000 compounds, right? In this broad drug repurposing hub for the sake of like the speed of experimental validation. But now what we can do is actually go beyond this set of compounds and go to like 10 to the 8th or even beyond. And by going to a larger collection of compounds, we actually found like 8 more compounds with various inhibition against 5 different bacterial species. And these are still in vitro results, not tested in mice yet. But we can already see that there are 2 compounds that has like broad spectrum antibacterial activity against all 5 of them. So in a way, this methodology, like based on deep learning, gives you it's quite flexible, and I believe there are a lot of new discovery that will come out in a variety of contexts.
And just to clarify, you still have only one model.
I still just have one model. That's even more amazing because
I just looked into training species-specific models where you can basically, train different ones and then see how do they compare, do they learn different things, etc.
Yeah, so we are still collecting many of the data. That's why we don't have the results yet. But even if you just train a model on E. coli, it can still provide something for the other bacteria species.
Yeah, but the converse, do you get any better by training on the other bacteria species, or there's no benefit to training in a sort of species-specific way?
We are still collecting data. That's why we don't have results yet.
Okay, sounds great.
Yeah. So lastly, I kind of mentioned that the graph neural networks are like better compared to other like traditional methods, right? So in this case, we want to do this retrospective experiment. If I have known that halicin is good, can other models also discover halicin? And it turned out that, for example, these feed-forward neural network based on this like a Morgan fingerprint, it does pretty bad. Like the rank of halicin is around like 1,000, right? We could only test around like 100 compounds back in the day. So in a way, none of these traditional methods, even some of them using neural networks, cannot discover halicin if we use these models. So in a way, it shows that these learned features are better than these hand-designed features using this simple experiment in terms of drug discovery.
Can I ask the converse? Are any of the top-ranked compounds from the other methods helpful at all? Basically, yes, the things that you discover, they rank pretty poorly. But what about the things that they rank highly? Number one, are they experimentally useful at all? And number two, how far down are they in your model? Is it that you guys capture completely different parts of the space, or they're still highly ranked in your model if they're highly ranked by their model?
Yeah, that's a great question. So what I remember was these traditional models, they just rediscover the existing antibiotics or things that are similar to the existing antibiotics.
Yeah.
For my model, we also rediscovered a lot of these existing antibiotics. That's why, or the something that's similar to the existing antibiotics. That's why halicin is only ranked at 61 because the model is a lot more confident with something that's similar to the existing one.
Got it. And now, in terms of the diversity of structures, I mean, it's very cool that halicin is a novel structure altogether. Are the 8 additional compounds that you mentioned in the same family, or are they completely different than halicin as well?
Oh, they are quite different. If you just look at the structure by itself, it has nothing similar to halicin. Just looking at the structures.
Yeah, and then the other question is, what about optimization? In other words, there's the sort of discovering some new sort of subsets of the space, and then there's optimizing within that subset. Is your model also good for optimization? Because it doesn't necessarily need to be the same model. Once you have, the where to look, you could develop a different model for the fine tuning. Have you looked into the fine tuning versus general where to land question?
You mean fine tuning the structure, right?
Fine tuning the structure, yes.
Yeah, I think that's what I will come in the end in the graph generation part, that I can learn to optimize the structure to increase certain properties.
Perfect. Because the equivalent that you would have is sort of the BLAST algorithm, for example, can tell you very rapidly a hit, but then you can use completely different methods to sort of make that better. And in the same way, maybe your method is amazing at sort of scoring, but not necessarily fine tuning. But it seems to be doing both. I'm just curious certainly the graph neural networks you're going to talk about afterwards can further improve it. But I'm just curious if you took like a different loss function that basically says for sort of fine scale differences, whether you will need another set of features or whether the same features actually do fine scale as well.
Yeah.
Thank you.
Okay, so let me go to, so this sort of concludes the first part. Let me jump into the second part, so if you look at the second part on like infusing biological knowledge into these graph neural networks, and I will demonstrate its application to COVID-19 drug combination discovery. So here, if you look at, so this is the graph neural network model that I just described, right? If you look at this model, it's still just looking at the structure by itself, right? The input is just the molecular structure, and you're sort of inferring some biological knowledge just from data. So in a way, that this model is losing a lot of information, right? Properties may depend on like additional biological information. For example, whether this molecule binds to certain proteins or biological targets, and maybe that's why that it killed the bacteria or the virus. In a way that this model does not tell you that much about the inner like mechanisms or how it actually arrived at the end property. So, and also when it comes to like viruses like COVID, which we don't have enough data, we actually need to bring back these biological knowledge into the model in order to generalize better. So that's sort of the motivation. And I will showcase, so basically I want to build some sort of model that are aware of certain biological knowledge. So that's the case study we did in COVID-19 drug combinations very recently. And the reason why we wanted to do drug combinations instead of single drugs is simply because they are more effective. So here, remdesivir is it's a FDA-approved drug for COVID-19, and it's only a single compound. But recent clinical trial shows that its mortality rate is not low enough, right? And it's only 4% lower than the placebo. So we need to find like better treatments. And if you look at like other viral infections, disease like HIV, you'll find that most of these HIV treatments are like drug combinations, right? Because they are often more effective. So this motivated us to find drug combinations for COVID-19. So here, what we want to do is to find drugs that are synergistic to each other. Here, I'm kind of being hand waving here, but basically by synergy, what I mean is if two compounds are synergistic, it simply means that 1 + 1 is greater than 2. Basically, if you have two of them combined, their effect is actually much greater than their additive effects if you apply them separately and just add their effects together.
But frankly, even additivity would have been fine, right? Basically, as long as they're not epistatic to each other, as long as the second one is still adding stuff.
It is still adding stuff, but how can I say? Yeah, you can, but this also has the cost of being more toxic and.
Yeah, I guess so.
Yeah. So yeah, I will. So in a way, we do want to reduce. So we don't want to overdose. Basically, that's why we want to have some synergistic compounds, because if two compounds are synergistic, then you can actually achieve the same effect with much lower dosage. Okay, so that's sort of the goal. We want to find drug combinations that are synergistic, and again, right, we can't really afford to do much experiments, and that's why we again wanted to do some sort of virtual screening, but this time on drug combinations. And the challenge here is, if you remember that for the antibiotic project, we had at least like 2,000 compounds. It's not really big, but at least able to train something. But here, COVID is a really new disease, and we really struggle to get any data. So at best, we found some sort of paper that they did a really small screening that had around like 100 or 200 drug combinations with their labeled like synergy. So this is certainly not enough, because graph neural networks are really data hungry, right? So in a way, we don't have this big data anymore, which neural networks really need. So instead, we have to add some knowledge into this neural network in order to generalize better, which can work well when we have just a small amount of data. So that's where the biological knowledge kicks in. And here, the knowledge that I wanted to add is the viral replication cycle. So basically, how can a drug block COVID-19 infection? So there has been a lot of work done by a lot of biologists studying like what's the viral replication cycle. I will not go too much into detail here because I'm not an expert, but basically there are three types of mechanisms. One is to block viral entry by inhibiting two receptors on the membrane. One is ACE2 and the other is TMPRSS2. These are the two receptors that the biologists have found to be crucial, that basically the virus takes advantage of these two receptors. And then you can try to inhibit the viral protease, like the 3-CL-Pro or PL protease, and so on. And lastly, there's also a lot of host targets, like host proteins that interact with the virus that's going on here. And there's a paper from UCSF which published a protein interaction map between the virus and the host, and we can try to inhibit these host proteins to prevent the virus from replicating. So that's sort of the basic knowledge that I wanted to add into the model. So that's sort of that leads to our model called combo nets, that try to incorporate both biology and chemistry. the difference between a combo net and the standard, graph neural network is in the molecular representation, right? So before, each molecule just have these learned fingerprints that's purely just encoding the structure of the chemical structure of the molecule. But now I want to add some additional knowledge, like what kind of biological targets that this molecule inhibits, and these are represented by these triangles that, in contrast with the traditional, in contrast to the previous chemical representation, represented by these circles, right? So now I can put both of these representation into the model to predict the antiviral activity of a drug. And by adding these, by modeling these biological interaction between the molecule and these biological targets, then we're able to bring a lot more data and can lead to better generalization. So this is sort of the three steps of our model. First, this model predicts the drug target interaction, like whether this compound inhibits certain biological targets. So here, what we do is we leverage a data set combined from Campbell, which is a public database with biological assays, and also from NCAT, from National Institute of Health. They both provided some data about drug target interaction in the context of COVID-19. So here, the data set is a sort of organized as a sparse matrix. Like you have these compounds and these targets. And here, 0 or 1 indicates whether this drug inhibits this target or not.
How much is known about COVID-19 in that database? I mean, do we know? I mean, because SARS-CoV-1, for example, in the SARS 2003 pandemic, is very similar. So were the different proteins of SARS-CoV tested against these chemicals?
So here, the NCATs, they just did a pretty small screen of these compounds against the viral protease of SARS-CoV-2. So they had some data,
okay,
but not that many.
And are you including human targets, or this is just virus targets?
So I'm including both the viral targets and the host targets, like Great. The HDAC is the host target.
Thank you.
Yes. Okay, so ideally, if this matrix is complete, then we can just directly plug it into the model as additional features. But here, because it is the matrix is too sparse, like because it is collected from different sources. So in order to fully utilize this information, what we need to do is to learn to complete this matrix by training the model to predict whether a drug inhibits a biological target. So then this matrix, it's like a matrix completion problem. By training on the existing data, the model can learn to fill in these missing entries. Then that's how we get this biological representation, the triangles on the top.
But in many matrix completion problems, you're assuming a lower dimensional representation will capture the actual matrix. How accurate is a lower dimensional representation? Namely, if you were to do dimensionality reduction on the matrix and do SVD or some other linear or non-linear dimensionality reduction, I'm just curious if there's like spikes in this matrix that will not be captured by a lower dimensional representation, or if in fact it's smooth enough that a lower dimensional manifold would do.
I see. Yeah, that's a great question. So here this is indeed a matrix completion problem, but we are not using these dimensionality reduction methods. Yeah. Instead, we are relying on this graph neural network to learn the representation for us by learning. So basically, I'm predicting whether this drug inhibits this like ACE2 or 3CL-Pro based on this graph convolutional network.
Perfect. Great.
Yeah, so in a way it is non-linear, and that's why it can capture these spikes. I mean, better than the traditional SVD-based methods.
Thank you.
Right. So this is the first step. and of course, I also have this learned representation of the molecular structure. That's the same as before. Now I have this two-part representation on the biology and also on the chemistry. Now I can use it to predict both whether a single drug is effective and whether a drug combination is effective. So here we are utilizing a single drug antiviral activity data from UNCATS, which they labeled whether certain drugs are antiviral against SARS-CoV-2. This is quite straightforward because it's the same, just I can put a feed-forward neural network on top of this representation and then convert that into a single numerical score, like basically the probability of this compound killing the virus. Now, the last part is to predict synergy for a set of, for two compounds, right? So now what we need to do is to first we can get this feature representation for drug A and B separately. So then I can learn to combine them into a single vector by doing this by this equation. And this sort of mimics the set inclusion exclusion principle that's motivated by this Bliss synergy score. So based on this single vector representation of the drug combination, I can then predict the combination antiviral activity. And this is a single numerical score. I can then compare this score with the other two scores, right? Basically, if these two, if PAB is much greater than PA plus BB combined, then it is synergistic, right? So I can train this model end to end using a handful of drug combination synergy data. So in a way that we are, even though this data set is small, we have this auxiliary data for drug combination, for the drug target interaction and also the single agent antiviral activity prediction. So that's how we bring different types of biological knowledge into this model to make predictions in the context of COVID-19. So this is sort of the performance of the model. I know there are quite a few like boxes here, but basically on the right is our best model that includes both the biological knowledge and the chemical representation. And if you just use the standard models based on like SVM or deep neural networks, without any of these biological knowledge, it just simply cannot generalize that well. And also, if you remove either the chemical or biological information, it will hurt the model a bit, quite a bit. So this hopefully gives you a sense of that, convince you that both biology and chemistry are important for the prediction task. So again, we don't want to just stop there. So that's why we wanted to actually make some predictions and test these compounds in some biological assays to verify whether the drug combinations are actually synergistic. So through the collaboration with the National Institute of Health and National Center for Advancing Translational Science, we tested around like 30 compounds in their UNCAT cell-based assays. And we actually managed to discover two novel drug combinations that are highly synergistic. So here are like two dose-responsive curves, matrices of these two drugs. One is remdesivir plus rezapine, and you can see here that I'm just sampling some numbers from this matrix. Basically, if you have the two drugs combined, it can kill the virus a lot better.
What about the zeros?
And this is similar here as remdesivir plus this IQ1S. So of course we need to, sorry, go ahead.
Why didn't you show us the zeros here? Is it that it's too extreme of a value or something, or?
Sorry, what? I missed that.
Do you see where I'm annotating on your slide, bottom left?
Here, yes.
So I'm just curious about those zeros, basically why you're focusing on.
Yeah. So I just wanted to make sure, because now if you have these two like combined, so here, right? So here, so basically I want to do a apple-to-apple comparison because I have this drug combination. It has two drugs. So in a way, the dosage is around like 6 micromolar, right?
Yeah.
And I need to compare with the numbers that is at least higher than 6 micromolar.
I see.
Yeah. Otherwise, I'm cheating, right? If I give you like 100 micromolar drug, then everything works.
Yeah.
But the human dies. So
got it. Perfect. Thank you. Very, very helpful.
Right. But of course, we still need to study these combinations in like human cells and also in animal models. So these are still like ongoing. But this is hopefully just gives you some idea of what the importance of bringing biological knowledge into the model. And now comes to the last part is de novo drug design. I will talk about this part is more on like machine learning in contrast to the previous two parts that had this. In this part, we don't have much biological results, but this is sort of the more ambitious goal of like solving this inverse problem. And this is important because if you look at the previous approaches, we train graph neural network, we rank all the compounds, and we do some experimental validation. It is good because it is fast, but it cannot scale up to the entire chemical space that I kind of pointed out in the very beginning, right? So we cannot do this virtual screening if we want to have like explore the entire chemical space. So that's why we need to do something totally different. And that's where the graph generation stuff comes in. And basically, if you look at drug discovery, it's actually a generative modeling problem, right? You want to learn a distribution whose mass is concentrated around these good molecules. Then if I can train this kind of generative model, then I can just directly sample good molecules from your model, right? Then life is good. But of course, so we can efficiently explore the entire chemical space by doing this efficient sampling. But it is a challenging problem because we need to now generate graph structures rather than these numerical numbers that standard machine learning models do. So I would like to skip some of the stuff. Is that okay? Because I know that it's running out of time.
I think we generally are okay with running a little over time. So I would prefer that you cover what you need to cover. And then if students need
well, it's only like around like 10 slides left. So
perfect.
I think we're good.
Take the time you need to cover it. We can go until 35 easily.
Sounds good. So, right. So there are two like prior approaches before what I did. First is this sequence-based method. So in chemistry, there's a sort of a representation called smile string. It's a domain-specific language. You can convert a molecule, a graph, into this 1D sequence. And once you do that, you can actually just directly apply recurrent neural networks to generate molecules, right? This is like very standard techniques that's widely used in natural language processing, and it's available in PyTorch or TensorFlow. It's very easy. But the problem is this string representation is quite brittle. If, for example, I have these two molecules or two graphs here that are almost identical, right? There's only like a little bit of difference here. But if you look at their smile string representation, they are quite different. And you see here on the, I highlighted all the differences in red, and it's a quite big chunk of them. So in a way, it is quite problematic, right? This representation, it maps, even though two molecules are similar, but they actually have quite different string representation. So in a way, it actually has a poor performance if you apply this to drug discovery. I will come to that later. Another previous solution is based on this node-by-node generation. It's also relatively straightforward. Basically, I just do this graph generation sequentially by adding one node at a time. So for example, I have this partial molecule here. I can predict a node, and then I can predict what's the atom type, and then I can also predict whether how this atom is connected to the existing atoms, right? So this is, and I can kind of do this procedure iterative. But the problem is these molecules are typically very sparse. So in a way, if you have like n nodes, you typically just expect to have like linear number of edges. But if you take this node-by-node generation approach, you actually need this like n squared complexity because in each step you need to predict n number of edges. And you need to do this n times. So when it comes to this, like when you apply this node-by-node generation to to drug discovery, you immediately find a problem. So a common approach is to use this variational autoencoder to generate molecules. You basically learn some sort of encoder that maps the molecule into this low-dimensional representation, and then decode it back to the same molecule. And you train this variational autoencoder to reconstruct the input molecule from its latent representation. But so a diagnostic test is to see whether this decoder can reconstruct a input molecule. If it couldn't, then obviously the architecture has some problems. So it turned out that this node-by-node generation has quite a bit of problem. If you look at the chart on the right, here the x-axis is the number of the atoms in a molecule, basically how large is a molecule. And the y-axis is the reconstruction accuracy. And you can see that the model kind of decreases significantly. For example, this COVID-19 drug called remdesivir, it falls in this range, and the reconstruction accuracy is already below like 40%. So this obviously is not good enough. This motivated me to think about some new architectures from for this graph generation problem. And node-by-node generation fails because it is designed for arbitrary types of graphs, and specifically dense graphs. So it might not be the best choice for like molecules. Given these challenges, I think it's helpful to think about what kind of inductive biases that we should incorporate, right? These recurrent or convolutional neural networks, they all leverage this inductive bias of like the input is a sequence or the input is like a grid. So that's why they're successful. So what kind of inductive bias do molecules have, right? So when you look at molecules, they typically are sparse, and they also have low tree width. And let me explain what that really means. So to explain this, I need to explain, I will just explain my model called Junction tree variational autoencoder that utilizes this low tree width prior of the molecule. So basically, what we can do is, so this method is inspired by the Junction tree algorithm in the classic like graphic models. And to leverage this low tree width of the molecule, what I do is I can decompose a molecule into a tree structure called junction tree. And what this junction tree does is, each node is no longer just a single atom. It is actually it can be a motif or a subgraph. however you call it. So it's like so because the molecules have low tree width, each motif is relatively small. So the tree width of a graph is basically the largest motif that you can get out of these tree decomposition steps. So here, this molecule has like tree width almost 5 because there's that's a 5-member ring, and that's the largest. And you can actually decompose rings further, but here we decided to just keep rings because they are chemically meaningful. So this is what I mean by having a low tree width, right? So if you do this tree decomposition, you actually end up having these motifs that are pretty small. And another aspect is if I apply this tree decomposition over a large collection of molecules, it actually doesn't result in that many types of motifs. I did this over like 250,000 different molecules or different graphs, and it turned out that there's only around like 600 motifs, different types of motifs. And these motifs have like over 99.9 coverage over a new molecule. So basically, there's not that much variety of these motifs. And that's because molecules have low tree width. So given these two observations, what I can do is I can design a new type of variational autoencoder that looks like this. So I can map a molecule into a continuous representation that incorporates this junction tree that builds on this intermediate junction tree representation. So I will embed both the molecule and the junction tree into this low dimensional vector, and then I can decode them back to the molecule. But I will first decode the junction tree and then decode the molecule. And this is advantageous because trees are much easier to generate. Let me explain why this is the case. So I will just run through the model architecture. Let's first look at the encoder. It is different from standard graph neural networks because we have both the molecule, both the graph and the junction tree. To be specific, I can first run a graph convolution in this graph, which learn like the feature vectors for each atom. Then I can propagate these atom vectors into the junction tree and run graph convolution there. So now each motif receives a motif vector that represents how these motifs are like connected together. Now, based on these encoded representation, now I can generate a graph in the following way. So in each step, I can the model decides. So it is also like a sequential model. In each step, the model decides which motif to add next, right? So basically, we need to pick what's the next motif then we need to add to the current molecule that we are expanding. So this is simply a classification task. Out of like 600-ish motif, I pick like this single bound, right? I can add this single bound into the molecule. Then I can predict how to attach this motif to the current graph by predicting what's the right attaching points between this motif and the existing graph. So in this case, we should attach this atom to this atom here. So then I can attach this motif to this graph, and this sort of gives you this one-step procedure, one-step expansion of the molecule, and I can repeat this process multiple times until the model decides to stop. And the reason why this approach is advantageous is because now you're generating it motif by motif. It actually requires the time complexity is actually linear to the because you are generating this tree instead of generating this graph. So generating a tree, you can actually do this in a linear number of steps. And as you can see here on the right, the red curve represents the reconstruction accuracy of this motif by motif generation model. And you can see that the reconstruction accuracy remains high if you have a large, even if you have a molecule like up to 100 atoms. I know this is not common for drug-like molecules, but this demonstrates the advantage of the motif by motif generation.
So Wengong, in practice, how often do these molecules have cycles? I mean, do we care about molecules that have cycles? Number one. Number two, are they not that common in nature because biological processes are just not very good at generating cycles? And number three, are they not very common because biological processes are not good at using cycles?
I believe that large cycles are hard, but the small cycles like benzene, I think it's really important.
No. If the cycle itself is part of your motif, that I'm not concerned about that. I'm just curious about your tree, whether the tree itself has a cycle, whether you could sort of loop back.
The tree does not have cycle because it is a tree.
But do natural molecules have cycles or not?
Here, so if you have this additional edge in between, then the junction tree will change into like 3 nodes. Each one is a cycle. Does that make sense?
It's only large cycles that you're not able to capture, basically.
It's only the large cycles that I was not able to capture, but in this case, it's not that common for drug-like molecules.
That's what I'm curious, but is it common for outside drug-like molecules? Basically, do plants generate, large molecules with cycles or not really?
Oh, they do. The natural products would have these large cycles.
Got it.
But again, I think from a mathematical perspective, this junction tree representation does not have to be, you can decompose the cycles. That's totally fine. But I just decided to keep it that way because the rings are quite meaningful in molecules. Perfect.
Thank you.
Right. So this is sort of the last slide that I have is on the molecular optimization. So it relates to the fine-tuning step that you mentioned. Basically, if I have some drugs that have like certain good properties, for example, I discovered helicin, but it may not be like drug-like enough. For example, this drug likeness encompass many like physiochemical properties, right? It greatly affects how it can be absorbed in the human body. So let's say that helicin is not good enough in terms of this drug likeness. Then I can perhaps learn to modify this compound a little bit, do some local modification so that it can improve the drug likeness, so that it can be better absorbed by humans. So this is where these generating models are good at, right? So I can not only generate molecules from scratch, but the model can also learn to modify existing graphs or existing molecules to improve its existing, to improve its drug likeness or some other physiochemical properties, while keeping its like antibacterial properties. So as you can, so we did this simple experiment. In this case, drug likeness is measured by QED, and you can just directly compute it. So in a way that every molecule that you generate, I can actually compute its QED score quite easily. So as you can see here, this sequence-based method can only do around this. So here the y-axis is the success rate. Basically, how often that you can make a low non-drug-like molecule into a good drug-like molecule. So here, this smile space or sequence-based approach, it does pretty bad, around 58% because of the wrong representation. this node-by-node based approach, if you move into this graph representation, it immediately does a lot better. But motif-by-motif approach can do even better in this task. So it shows also the advantage of this motif-based generation. So this is sort of the three parts of the story. I hope that it gives you a little bit of flavor of what we can accomplish in this space. And just as the last slide of the talk, there's a really big push in deep learning for molecular sciences. There's not only drug discovery, but also organic chemistry, biology, or material science. So in a way, there's really a lot of exciting opportunities here. And with that, I would like to thank all my collaborators. All the works that I've done presented in today's talk is not really, it's really a teamwork. So I really appreciated all the professors and the students who helped me on doing these experiments, and also the government agencies and pharmaceutical companies who really helped give suggestions and feedbacks. Yeah, with that, yeah, that's all of my talk. Thanks a lot.
Thank you so much. Can you go back to your last slide, the one about the outlook? Because I was wanting to say a few words about each of those. So you told us a little bit about drug discovery, but if you want to tell us a little bit about what's your outlook for each of these areas, if you can say, well, here, I see the main challenges that is X, Y, Z, etc. I would love to hear your take on chemistry, biology, material science, but also drug discovery and a little bit of your view for where the field is heading.
Right. Yeah. That's a great question. So I can talk a little bit about the biology a bit more, because here there's something quite unique. For example, I mean, definitely you guys have heard about AlphaFold, which is exciting progress in protein folding. So in a way, when we in today's talk, I'm mostly just dealing with these small molecules, but for proteins, these 3D structure is actually really important, and it requires a different skill set, different types of techniques. Not just like these operating on graphs, but now you have to deal with these geometric objects in 3D space, and how you design deep learning algorithms that can efficiently extract or learn representation out of these 3D structures is something is very interesting. And also in material science, you have to deal with large structures like these polymers. These are gigantic. So how to scale these deep learning methods to these large structures is also a big open question. Yeah, I think in terms of drug discovery, so far I have not yet bring in. It's more like ligand-based drug design, right? I only look at the molecule, and I had some work on bringing protein or biological targets, but have not yet did bringing the protein structure, the 3D structure, into the drug design, right? When you talk about how the ligand docks into the protein, we obviously want to model that the protein ligand complex rather than just looking at the ligand. So there are just tons of opportunities. I'm just mentioning these that sort of immediately comes up in my head. Yeah.
Can you tell us a little bit about chemistry as well?
Yeah. So for chemistry, it's more about how you can synthesize a molecule, right?
So the actual series of reactions, basically.
Right. How you can cook up a synthesis plan for a new molecule that's designed by some generative model, right? So these are some of the work that I've been doing, but definitely it's not just me, but really this community, a lot of brilliant people are working that in all these problems. So yeah, just want to give this outlook so that people can go into these papers to see what's in there. Yeah.
Is that the same Jin each time?
Huh?
Is that you each time?
These are my work. Yes. These are all done by me. Just but it's a big field. A lot of people are doing that. Yeah.
Well, that's really wonderful. Anyway, we are privileged to have you here. So thank you so much. This was a fantastic lecture. And I certainly learned so much, and I hope the students learned so much as well. So thank you. Congrats.
Thanks a lot. Yeah, let me stop share. Yeah.
Awesome. Everybody's super thrilled. So thank you, thank you, thank you. And then looking forward to interacting again because I have so many more questions. I guess I'll be reaching out.
Sounds good. Yeah, thanks a lot for inviting me. And it's my pleasure to be
One
here.
question that we ask to all the guest lecturers is if students are interested in doing projects in this space, would you be able to provide some mentorship for them?
Sure. Yeah, sounds good.
Wonderful. Okay, awesome. Thank you so much.
Yeah.
TAs to please stick around. Thanks, Wengong.
Okay, bye-bye.
Bye-bye.